Hi, it is our privilege to present to you our paper entitled Molecular Identification of Cone Snails from Family Conidae Gastropoda at Verde Island, Batangas, Philippines using mitochondrial gene markers Cytochrome Oxidase 1 and 16S rRNA. I am your presenter, Rotella P. Payawal from the Polytechnic University of the Philippines, Department of Biology. For the introduction, 85% of family Conidae is genus Conus, with 803 known species according to Worms of 2014. In terms of diet, they are vermivorous, piscivorous, and mollucivorous. They are also predatory and venomous in terms of their habits. They are pharmacologically, hence economically important because of their conopeptides, which are used into drugs. And they have very intricately patterned shells. These patterned shells well loved by collectors. For the mitochondrial gene markers that we used two genes, CO1 and 16S rRNA. They are mitochondrial genes, and the next question is, why did we use mitochondrial genes? Because mitochondrial genes are maternal genes, hence they are highly conserved. For the CO1 mitochondrial gene, or the cytochrome oxidase 1, they have 650 base fragment. They are the official barcode for animals according to CBOL, or the Consortium for the Barcode of Life. They are very easy to amplify for the broad spectrum of phyla, and they have a rate of nucleotide substitution which is quite high, and there are at least 3,000 publications which has most extensive use in the DNA barcoding of animals, and they're highly conserved with very low changes of gene sequence over time. Okay. Now for the 16S rRNA, they are non-coding gene. These are the rRNA transcript that forms part of the mitochondrial ribosomes, which plays an important catalytic role during translation of messenger RNA into proteins. They are one of the slowest evolving genes, which are rapidly evolving regions, and these evolving regions are the control regions, and they have a combination of variable and conserved regions within the same gene, which are very popular among scientists. Now, for the objectives of the study, generally, we want to assess the species of family Conidae present at Verde Island, Batangas, Philippines using molecular methods. And also, specifically, we wanted to identify the species using the mitochondrial gene marker CO1 and 16S rRNA. Also, to generate a species list of cone snails at Verde Island and establish such record for future monitoring and conservation efforts. Now, also, to reconstruct eventually the molecular phylogeny of the species collected using the generated DNA sequences. For our methodology, let me introduce first our study area. If you can see here, this star is where Verde Island is. This is our sampling area. And the Verde Island is within the Verde Island Passage. The Verde Island Passage is known to be the center of marine biodiversity in the planet, which is actually located at the tip of the coral triangle. The coral triangle is at the center, or at the central Indo-Pacific region, covering parts of Indonesia, Papua New Guinea, Malaysia, and the Philippines. And this is the Verde Island Passage is the part of the coral triangle in the Philippine side. What are my collection sites? I have at least 6 barangays. San Agustin Canluran, which is made of Sitio Mahabang Buhangin and Sitio Gabihan, and also Sitio Sampalukan. We have San Agustin Silangan, San Antonio, Liponpon, San Andres, and also San Agapito. San Agustin Canluran have generally sandy and rocky substratum, but there is also a muddy part. While generally sandy and rocky are these 3 barangays here, and this is 4 barangays, and this last barangay, San Agapito, is a sandy area, which is basically a swimming area and a tourist area at that. Now, for the methods that we used, we preserved, and we identified by collecting cone snails at the intertidal areas only up to 10 meters deep. and we did not include the deep sea species, and we preserved the cone snail tissues at 95% ethanol. And we extracted the DNA, we amplified using PCR, of course using the primers for CO1 and 16S rRNA gene markers, and we had the DNA sequencing. And after it was sequenced, it is now time for the phylogenetic analysis. First thing we did is a sequence alignment, then analysis through BLAST, and then construction and analysis of phylogenetic trees. Now for the results, analysis, and discussion, this is the analysis in the NCBI BLAST of our samples. For the maximum identity score, this is, we compared the identity of our samples with the one that is present in the data of the NCBI. The CO1 yield 95 to 99%, while 16S rRNA had only maximum identity score of 81 to 99. 38 species had successful identification out of the 45 phenotypes that we gathered, while from the 38 we only have distinctly identified 21 species. Now, if you notice, there is the maximum identity score of 16S rRNA is a little bit lower than CO1. Now, for the molecular identification of species collected, comparing CO1 and 16S rRNA genes, 26 samples had matching identities in the two gene markers, meaning both of them reveal the same identities in a species. Now, 11 samples had differences in identification. No DNA samples were extracted from 6 phenotypes. And Conus miles, the one that you can see on the left, was only identified through CO1, and Conus immachatus here on the right was only identified only through 16S rRNA. Now, what are our significant findings? Both genes were successful in identifying the Conidae species collected. Some differences in the identification were observed based on the phylogenetic trees that we generated from phylogenetic analysis and the pairwise distances that we generated from the BLAST results from NCBI. And there were disagreements with identification, of course, that you have seen earlier, but it coincides with the lower identity match scores in 16S rRNA that I have mentioned in the previous slide. And the concatenated phylogenetic analysis. What is a concatenated phylogenetic analysis? This is a phylogenetic tree that combined the trees both from 16S and CO1. And the identification from both when they were combined follows the signal and is usually confirmed in CO1, which of course coincides with because of the lower identity match scores in 16S rRNA. Now for my conclusion, the mitochondrial markers are successful in the identification of the Conidae species collected from Verde Island, Batangas, Philippines. The phylogenetic analysis show consistent clades, and well-defined lineages, and the species listing of Conidae species were generated and records are established. Unfortunately, the phylogenetic analysis were identified, done through the phylogenetic trees, and because of the requirements in the number of slides, I was not able to present those phylogenetic trees. Now, for my references, they are written in the paper we submitted to the organizer. Thank you, and this is the end of my presentation. I hope you enjoy.